compounds from their hosts. Hemiparasites still have leaves with stomata and rather ordinary stems. Their roots, however, penetrate the epidermis or bark of host plants rather than soil. Holoparasitic plants have neither chlorophyll nor photosynthesis. All of their ATP is produced by aerobic respiration of glucose obtained from the host plant, and they probably need little reducing power. In all cases, after the ancestral parasitic plants became holoparasitic and lost any need for photosynthesis, mutations that curtail leaf development became beneficial. Most holoparasites have either small, scale-like leaves or none at all. Daughter is one of the few holoparasites that has any ordinary plant-like features left in its body. It has long, slender stolon-like shoots with tiny leaves. It twines around host plants, inserting haustoria into them and then growing onward. Its plant-like features of nodes and internodes seem to be necessary for it to spread from branch to branch of a host or from one plant to another. Table B2, 1 families that contain parasitic plants. Most holoparasites are subterranean. Their seedling roots invade host roots, then the parasite's embryo develops into a rhizome-like shoot with small, tough, protective, scale-like leaves. Being underground, it is invisible to herbivores, and temperatures and humidity are more stable, less variable than above ground. Many photosynthetic plants also have rhizomes, but many of their axillary buds grow upward as aerial, chlorophyllous branches that never occurs in holoparasites. A few of their axillary buds grow above the soil surface as inflorescences, displaying flowers to pollinators. In other holoparasite species, development is even less like that of ordinary plants. The subterranean rhizome has no leaves at all, no axillary buds, nodes, or internodes. The growing apex consists of a mass of meristematic cells but has neither leaf primordia nor a root cap. Internally, the rhizome's tissues are not typical of either root or shoot, having neither a single ring of bundles typical of a stem nor a central mass of xylem as ordinary roots do. Even the surface is just a rough, irregular proliferation of cells rather than a smooth sheet of epidermis. The term runner is often used to indicate that it is not an ordinary plant organ. Several holoparasitic plants live entirely within the body of their hosts. After their seedling root penetrates the host, the rest of the embryo dies. The roots grow deeper into the host, penetrating its cortex, phloem, xylem, and pith, and they make up 100% of the parasite's body. The parasite has no shoot at all. The term roots is in quotation marks because these structures have no features typical of roots. Technically, this is an endophyte, a plant that lives within another. The endophyte of the mistletoe Tristrix aphyllus, which grows inside the cactus Trichocereus chilensis, consists only of a branching web of uniseriate filaments of parenchyma cells. After some months or years, the filaments become multiseriate as their cells undergo longitudinal divisions. At some point, a bit of phloem develops and an occasional, isolated vessel element. The entire plant body has no roots, stems, leaves, epidermis, cortex, pith, and only a few cells of xylem and phloem. Detailed studies are needed, but it appears that every surface cell of the endophyte body absorbs water, minerals, and organic molecules from the host's tissues. With the entire body being absorptive, there is little need for conduction. When stimulated to flower, parenchyma cells just below the host epidermis proliferate into a nodule of callus, an adventitious inflorescence apical meristem forms, and then it breaks its way through the host epidermis. Because it has an ordinary apical meristem, the inflorescence has epidermis, cortex, a ring of vascular bundles, and pith. Endophytes of Viscum minimum, Pilostyles thurberi, and several Rafflesia species are similar. After a green photosynthetic photoautotroph has produced glucose, it then synthesizes all other organic compounds needed for its structure and metabolism. It may be that some holoparasites do the same thing. They may merely obtain glucose from their hosts and then use various types of respiration and anabolic pathways to synthesize all other organic molecules. It would not be surprising, however, to find some species of parasitic plants that have become dependent on their hosts for at least some amino acids, lipids, or vitamins. Despite being so modified, the endophytic bodies of holoparasites are healthy, thriving, and well adapted to respiration-based metabolism in a unique environment. Holoparasitic plants show us that many features of ordinary plants must be very expensive and risky. After a plant has an alternative to photosynthesis, ordinary plant features are no longer adaptive, and mutations that prevent their development are advantageous. On the other hand, for plants that do photosynthesize, plant-like bodies are worth the trouble. Figure B2, 2a. The slender orange stems are Cuscuta, a holoparasite that still has leaves and stems, but its inner nodes are extremely long and slender and its leaves are tiny. It is producing adventitious roots that develop as haustoria, penetrating to the vascular tissues of the host, Justitia. Figure B2, 2b. Prosopancha americana is holoparasitic, and most of its highly modified body remains underground. It has no need for photosynthesis, obtaining all its water and nutrients from the roots of its host. This flower is also highly modified. The black and white structure is a massive set of fused stamens. Figure B2, 2c. The small cluster of cells with large red-stained nuclei are part of the body of Tristrix aphilis, a holoparasite whose vegetative body grows entirely within the body of its host, in this case, the cactus Trichocereus chilensis. The body of T. aphilis consists of just parenchyma and lax leaves, stems, epidermis, and virtually all other features of an angiosperm body. Figure B2, 2d. The dark blobs do not belong to the visible shoot. Instead, they are the flowers of a holoparasitic plant, Pilostyles thurberi, growing completely inside its host, the shrub Dahlia. Holoparasites typically must emerge from underground or from inside their hosts when they flower. Figure 2, 21. Simple diagram representing the raw materials and products of photosynthesis, carbohydrates produced by photosynthesis are subsequently converted to many other compounds. Respiration is another type of energy metabolism, and it occurs in all organisms, with no exceptions whatsoever. As just mentioned, glucose is used to store energy and to move it from organ to organ. But a problem with glucose is that it stores too much energy for certain needs. Cells force many metabolic pathways to run by adding a small bit of energy to 2 or 3 steps of the reaction. A molecule called ATP provides just the right amount of energy for most of the steps. Glucose has about 36 times as much energy as is carried by ATP. Respiration breaks down glucose and transfers its energy to ATP. During photosynthesis, light energy is captured as carbon dioxide combines with water, producing oxygen and glucose. Respiration is more or less the reverse. Oxygen and glucose react, producing carbon dioxide and water, with the energy being transferred to ATP rather than being given off as light. Plants also need minerals such as nitrogen, phosphorus, potassium, iron, and others. Plants use these in much the same way we do. Phosphate is needed for ATP, nitrogen becomes part of amino acids and nucleic acids, iron is attached to molecules that transfer electrons, and so on. Also, like us, plants are not especially effective at recycling minerals. We lose minerals as we sweat, urinate, and defecate. Plants lose minerals as they shed their leaves, withered flowers, and fruit. They pack their seeds with minerals that the seedlings will need, and all those minerals are provided by the parent plant. The amount of minerals a plant needs is surprisingly small. The ash that remains after a fire contains all the minerals that had been present in the wood. Figure 2, 22. Only the leaves of these palms are green and photosynthetic. They are the only parts of the trees that produce sugar. All other parts, such as the long stems and the underground root systems, have no chlorophyll and must obtain all their energy in the form of sugars transported downward from the leaves by phloem. Plants are master chemists. Starting with just glucose, minerals, and water, they synthesize every single molecule they need for all aspects of their life. They synthesize all amino acids, nucleic acid, and fatty acids. They make all the pigments of flowers and fruits. They make waxes, oils, resins, and fragrances. And poisons. Many plants defend themselves from pests with an amazing array of poisons. Some are bitter or irritating, others kill animals quickly. Some of the most toxic chemicals interfere with cells in the brain and nervous system. Although we humans need a complex diet that includes several amino acids, fatty acids, and vitamins that we cannot make for ourselves, that is never true of plants. Their balanced diet is sunlight, carbon dioxide, water, and minerals. Figure 2, 23-adenosine triphosphate. Breaking off the last phosphate to produce adenosine diphosphate results in a more stable set of electron orbitals, and energy is given off. The same is true of removing the second phosphate, but not the third. Plants and people. Box 2 to 3 toxic plants. Consider 2 facts. Plants live among herbivores, and the world is full of uneaten plants. What is the cause? Many plants produce toxic compounds. Poison ivy, death camas, poison hemlock, and deadly nightshade are familiar examples, but the full list of toxic plants is very long. Many plants we think of as being non-poisonous are only harmless because we stop eating things that are bitter or that burn our mouths. Few people would eat enough chili peppers to get a lethal dose. The following are some toxins in plants we live with. Fascinating and well-illustrated accounts of many are given in Handbook of Poisonous and Injurious Plants by Lewis S. Nelson, Richard D. Shea, and Michael J. Ballack. Figure B2, 3. Each leaf of poison ivy has 3 leaflets and the defensive compound urushiol. Anticolinergic poisons. Many nerve cells have receptors that detect the presence of a small molecule called acetylcholine, which causes a reaction in the nerve cells when it binds to the receptors. Plants such as Atropa and Datura produce chemicals that bind to one of these receptors, preventing acetylcholine from stimulating proper nerve transmission. Nicotine-like alkaloids. Alkaloids are small molecules that contain nitrogen and somewhat resemble amino acids. Alkaloids of conium, nicotiana, and sophora block another acetylcholine receptor. Symptoms occur in the parasympathetic and sympathetic parts of the nervous system, the brain, and the junctions where nerves attach to muscles. Convulsant poisons. The small tree strychnus produces strychnine, which causes motor nerve cells to be hyperexcitable and results in convulsions. Several other plants produce convulsants. Capsaicin. Chili peppers such as habaneros, jalapenos, and tabascos in the genus Capsicum contain capsaicin, which induces some of our sensory nerve cells to release a chemical called substance P, which specifically stimulates other nerve cells that normally respond when we are being burned. Artificial selection by humans has resulted in chili peppers that range from having almost no capsaicin to some that are dangerous. Capsaicin is hydrophobic, and thus, drinking water will not cool your mouth. However, the fat in cheese, butter, and milk will absorb it and give some relief. Cardioactive compounds. Digitoxin and digoxin and digitalis inhibit a set of proteins that use the power of ATP to pump sodium and potassium across a membrane. When this pump is blocked in heart cells, it leads to an increase in the concentration of calcium within the cells. Although foxglove leaves are injurious if eaten improperly, these chemicals are used medicinally to slow and strengthen heartbeat. Cyanogenic glycosides. These are non-toxic in the plant, but enzymes in our digestive system cleave them into 2 parts, a sugar and cyanide. Cyanide inhibits the final step in aerobic respiration, preventing generation of ATP. These toxins are found in some of our most common fruits, malus and prunus, but in parts that we do not eat such as apple seeds or peach pits. Poisoning most often occurs when apricot seeds are used as components of herbal medicines. Mitotic inhibitors. Microtubules create the spindle that pulls chromosomes to opposite ends of the cell during nuclear division. The alkaloid colchicine in autumn crocus, Colchicum autumnale, prevents the assembly of microtubules. Cells exposed to this poison cannot form a spindle, and division fails. We humans have many tissues with rapidly dividing cells, such as the lining of our digestive tract, bone marrow cells that produce red blood cells, and seminiferous tubules in men. Catharanthus roseus produces the alkaloids vinblastine and vincristine that also block cell division. These are used in chemotherapy to stop cancer cells from dividing. Toxolbumins. These proteins bind to ribosomes and prevent them from translating messenger RNA into protein. Ricinus produces the toxalbumin ricin, one of the most toxic substances known. Jatropha species has a higher concentration of a related toxalbumin, and a single seed can cause severe poisoning. Urushiol. About half of all people are allergic to urushiol, an oily substance in species of Toxicodendron. All parts of these plants can trigger an allergic reaction. Remember that these plants do not have toxic compounds in order to protect themselves, or with the purpose of keeping herbivores away. It is just that toxic or injurious plants are more likely to be left uneaten compared with non-toxic plants and therefore are more likely to produce more seeds and more progeny. Table 2, 3 chemicals that plants produce starting with glucose. Overview of information in plants. A universal feature of all known life on Earth is that DNA is used to store information. Just as with energy and matter, organisms take in information, store it, move it from place to place, transform it, use it, and then pass it on to their offspring when they reproduce. The greatest source of information for any individual is its parents, the DNA contained in the sperm cell and egg cell that initiated the life of the individual. In plants, DNA is stored in the nucleus of each cell, as well as in plastids and mitochondria, which are also present in all living plant cells. When a cell needs to use some of the information, it transforms the needed information by making messenger RNA, and then it transfers that information to the area of the cell where it is needed. Most of the information is further transformed as the messenger RNA guides the production of specific proteins. Smaller pieces of RNA, often called microRNA, act by binding to various components of the cell, activating some, inhibiting others, and in general fine-tuning the activities of many genes. The DNA of each species is unique. For example, cherries and peaches have almost identical DNA, but the information in several genes in one differs just enough from that of the other so that cherries are small, red, and shiny whereas peaches are large, yellow, and fuzzy, and each has distinct flavors and aromas. A plant, just like any other organism, also uses information from the environment. Environmental information must be perceived then transferred to where it is needed, and finally the plant must respond appropriately. While the plant is still nothing more than a dormant seed, the environmental information it responds to is often very simple. It is warm enough to germinate. As temperatures climb from winter to springtime, at some point they are warm enough and many seeds then respond by becoming active, respiring more rapidly, and germinating. Seeds of other species remain dormant because they are dry, not cold. They need to detect when their surroundings have become wet enough to support a growing seedling. The environment also supplies a large amount of more complex information, in many forms. Sunlight provides the energy for photosynthesis, of course, and it also indicates the direction up from down. In all parts of the world except near the equator, sunlight provides information about seasons. Days are shorter in winter, longer in summer. By detecting this, a plant measures the season and responds by flowering and fruiting at the correct time and by becoming dormant in autumn. Roots are not exposed to sunlight, but they detect gravity and thus know which direction is down. Deeply buried bulbs also detect gravity and respond in two opposite ways. Roots follow gravity downward, whereas shoots grow upward. We are now learning that some plants are much more perceptive than we ever suspected. Tendrils bend toward anything that gives them even the slightest touch. Leaves detect the growth of a single fungus spore on their surface, and some orchids detect whether a pollinator has merely landed on one of their flowers or if it has actually taken the pollen. Figure 2.24. All plants here obtain energy from their environment, but think of the two trees. They both obtain information about which way is up. For the grasses in the pasture, up is in the direction of the brightest light, but for the tree on the right, the brightest light is away from the larger tree that is shading it, so bright and up are not the same thing for it. These trees also need to detect the approach of winter and abscise their leaves while they can, and they probably detect the presence of many insects and fungi as well. Figure 2.25. When these bulbs became active and started growing, all parts were so deeply buried in soil that they could not detect any sunlight. But both new roots and new leaves detect gravity, with roots growing downward, leaves growing upward. Various parts of a plant itself create information that informs the rest of the plant of their activities. Healthy leaves produce hormones that are carried to the roots. A large amount of hormone indicates the leaves are active and need water. The roots respond by growing and absorbing water and nutrients. In autumn, as leaves prepare to be shed, they stop producing the hormone, so the amount of the hormone decreases, indicating the leaves no longer need water. Roots respond by slowing their growth and conserving their energy. After a flower is pollinated, the developing fruit sends signals indicating that it needs nutrients for itself and the embryonic seeds inside. This communication among the parts of a plant is necessary to keep the plant integrated and all parts developing in coordination. It is especially remarkable considering the long distances involved between the thousands of leaves, flowers, branches, and roots of a large tree. Overview of plant diversity and evolution. All the thousands of species of plants of the world are not just a hodgepodge of randomly associated features. Instead, there are several large, recognizable groups such as mosses, ferns, conifers, and angiosperms. Within each group, we are likely to find certain features and unlikely to find others. For example, mosses never have xylem or phloem and they never have seeds either. As a second example, ferns always have vascular tissue but they never reproduce with seeds. Several aspects of the groups in Table 2.4 are not quite accurate, but the groups and terms presented there have been used for more than 100 years, and you will still encounter them often in books about gardening and roadside flowers. An overview of them will be given here. Definitions of important terms are grouped together in Table 2.5. Mosses, liverworts, and hornworts are often grouped together and called bryophytes. They are similar to each other because they not only lack vascular tissues and seeds, but also because plants in all three groups are haploid. Mosses, liverworts, and hornworts are unique among plants because they live their lives with only one set of chromosomes, not two. Their bodies are very simple, not much more complex than the algae that were the ancestors of plants. Table 2.4 Overview of the main clades of organisms. Table 2.5 Terms used for plant groups. Names in formal classifications are capitalized but informal names are not. The informal names of some of the groups here are angiosperms, conifers, cnidophytes, and mosses. Their formal names are Angiospermophyta, Coniferophyta, knee-tails, and Musci or Bryophyta. All other groups of plants have vascular tissue and are diploid. Many vascular plants resemble bryophytes in being unable to form seeds. They reproduce using spores and are typically called the spore-bearing plants. These are often referred to as ferns and the fern. Allies. An old way of thinking about plants is that ferns and fern allies had advanced above the bryophytes as they evolved to have vascular tissue, but then their evolution stagnated while other vascular plants continued to advance evolutionarily to the point where they could make seeds as well as vascular tissues. The more advanced seed plants then divided into two groups, those with naked seeds and those with clothed seeds. Here, Clothed seeds refers to seeds formed inside a fruit in contrast to naked seeds that are produced in a cone. The term naked refers to the time when a pine cone is extremely young. At that point, its scales are far enough apart that you can look in and see the individual naked ovules. The carpels of angiosperms are always sealed, and the ovules are never visible without cutting the young fruit open. There are other differences between gymnosperms and angiosperms that make us think of angiosperms as being more advanced. Angiosperms have more complex wood and leaves, as well as several modifications that improve photosynthesis. Angiosperms also cooperate with animals that pollinate their flowers and distribute their seeds, whereas conifers never rely on animals for pollination, and only a few cooperate with animals for seed dispersal. Figure 2, 26 mosses cover the rocks in this stream in the Olympic National Park. Mosses have no xylem or phloem and cannot conduct water very well, so they are typically very small plants. Although we associate mosses with damp places, many are adapted to deserts, being inactive except on rare days when there is rain, fog, or dew. Figure 2, 27. These are plants of the leafy liverwort Bassania trilobata. As with mosses, the bodies of leafy liverworts consist of a stem and leaves, but never any roots, xylem, or phloem. It is usually necessary to have a hand lens or magnifying glass to distinguish between a leafy liverwort and a moss. Figure 2, 28. This is a rather common thalloid liverwort that is easy to recognize. Its bodies never have any hint of leaves or stems, they are flat and about 1 centimeter wide, and they occasionally fork into 2 growing points. Their upper surface has a distinctive diamond-like pattern that resembles reptile skin. Figure 2, 29. Hornworts such as the Spheoceras laevis are widespread, but they only thrive when it has been rainy for a while. They grow in shady places, sometimes on bare soil, sometimes mixed among grasses. Each long projecting cylinder is the horn and is actually the diploid portion of the plant. Each horn is the same phase of the life cycle as is a tree, bush, shrub, or any other familiar plant. The small flat patches of tissue in contact with the soil are the gametophytes, the haploid part of each plant. It is the part that is similar to mosses, leafy liverworts, and thalloid liverworts. The life cycle of plants is much more complex than that of animals, but is fascinating. We can arrange the different groups in a linear sequence that seems to reflect plant evolution. The earliest true plants were more advanced than algae because they had simple tissues and organs but little else. This would be the moss level of evolution. Other plants advanced to the level of having vascular tissues, others continued evolving to the point of having seeds. Then one group evolved more than all the rest, to the point of having flowers and fruits. Some would even refer to angiosperms as the pinnacle of plant evolution. In this method, emphasis is placed on the level to which a group has evolved. The technical word for level is grade. We are grouping plants into grades, with the lowest grade being nonvascular plants, the highest grade being angiosperms. We still encounter this concept of grade when people refer to the lower plants and higher plants. This grouping by grade is very handy if you are not too worried about accuracy, but we now have a newer method of grouping plants, one based on groups that consist of all the species that evolved from a common ancestor. Such a group is a clade. Fortunately, grouping plants into clades gives us almost the same result as grouping them into grades. We need to abandon only a few of the old terms. For example, we no longer use the term gymnosperm. We suspect, but are not at all certain, that seeds evolved only once. If so, then all seed plants alive today descended from that original species of seed plant, and we say it was the common ancestor to all seed plants. Some of its descendants evolved into cycads, others became ginkgos, another group evolved into conifers, and still another group became angiosperms. There are 2 important points here. The first is that a clade consists of an ancestor and all its descendants, so the group seed plants is acceptable, it is a clade. But the word Gymnosperms refers to all seed plants except Angiosperms. It includes the common ancestor of seed plants but only some, not all descendants, so Gymnosperms is not a clade. We prefer not to use the term gymnosperm if we care about being accurate. Figure 2:30. This Selaginella lepidophylla is used as an example of a fern ally because of its interesting biology. It is a resurrection plant. It is a diploid vascular plant with xylem and phloem, and it grows in harsh deserts with long dry periods. This photograph shows the plant after autumn rains, when the plant is hydrated, healthy, and active. Compare with Figure 2:30b. This is also Selaginella lepidophylla, but after a long dry period, the plant has curled up, minimizing the amount of surface exposed to dry air and UV light. Although it looks dead, it is not only alive but very healthy, merely dormant, somewhat like a dry seed that is alive. After a rain, these plants would uncurl and look like those in Figure 2:30a. If forcibly uncurled now, the inner parts would be bright green. The second important point is that the ancestors of any clade of living organism all lived in the past, and for clades such as angiosperms or conifers or seed plants, the ancestors lived in the very distant past. Consequently, we can never be completely certain which ancient, extinct group was the common ancestor of any modern clade. Instead, we have to study the living organisms and try to infer the features the common ancestor had. Using angiosperms as an example, all modern as well as fossil angiosperms have vascular tissues, they produce flowers and they make seeds, they never make anything similar to a pine cone, and they are never haploid like bryophytes. From this we assume that the common ancestor of all flowering plants was a diploid vascular seed plant that made flowers but not cones. At present, we are able to analyze the sequence of nucleotides in the genes of organisms, and at present we have sequenced all the genes of many species. This provides an abundance of new information for constructing and testing our theories and models. Figure 2:31. These plants of Equisetum telmatia are diploid vascular plants that are now considered to be part of the ferns. These are descendants of an ancient group. Fossils of plants that have similar features can be found in rocks as much as 150 million years old. Equisetums are called horsetails or scouring rushes, and are distinctive in having a very coarse surface. They can be found almost everywhere, along streams and along railroad tracks in moist areas. Figure 2:32. This is a single plant of sword fern. It has a short vertical stem that supports the many leaves, and below ground are many fibrous roots. Ferns are diploid and have xylem and phloem, but their spores grow into tiny haploid plants that resemble the bases of hornworts. This large sword fern pictured here is the equivalent of a hornwort horn. In rainy areas of the Pacific coast of the United States, sword ferns are so abundant they resemble meadows of grass. Indeed, millions of years ago, before grasses evolved, most grasslands were actually huge patches of ferns. Ferns and dinosaurs were abundant at the same time, long before grasslands existed. The herbivorous dinosaurs ate ferns and fern allies. Figure 2:33. This cladogram is a hypothesis about the ways in which modern plants are related to each other. At the very base would be the ancestral plants, not much different from algae. Many years later, at B, some plants began to evolve into liverworts while the rest of the plants continued evolving other features. At C, some plants began to evolve into mosses, and so on. At some point between D and E, vascular tissue evolved in the main group of plants, and all plants that descended from these early vascular plants still have vascular tissue as well. That means that the xylem in ferns resembles the xylem in flowering plants not by accident but because both evolved from the same ancestral xylem. The lines that show the ancestry of cycads, cnidophytes, ginkgo, and conifers all start from the same point. That means we do not yet know which diverged earliest, and we do not know which group is most closely related to which other group. The line for the ancestry of angiosperms diverges a bit later, but it could be that this is incorrect and we should draw it coming from G, the same point as the cycads and others. The old concept of grades assumed that cycads, ginkgos, and conifers were evolving more slowly than angiosperms and, given enough time, they would finally at long last evolve up to the level of having flowers. Similarly, maybe one day in the future, ferns would evolve to the point of having seeds, and so on. It is like moving from grade to grade in school. Everyone starts out in the first grade and then all move along toward graduation, some moving faster than others, some stopping completely. This is the second problem with grouping plants into grades. It assumes that all plants have the same goal, that having vascular tissue and seeds and flowers is the optimum way a plant can be adapted to its environment. We no longer believe this. We now realize that there are multiple ways for an organism to be adapted to its environment. This is reflected in the questions: what are the alternatives, and what are the consequences? Consider mosses and angiosperms, two groups at the opposite end of the old grade system. Mosses are tiny and simple, whereas most angiosperms are large and complex. But mosses are very well adapted to their environment. They thrive in low light, on brick walls or bare stone. They need only enough sunlight, water, and mineral nutrients to make a few spores, and then they can reproduce. In the moss clade, evolution by natural selection has found solutions to survival that work well for small, simple plants. Having vascular tissue and seeds and so on would not make a moss any more adapted than it is without them. This is a crucial point I will emphasize many times. There are many, diverse ways to be adapted, numerous ways of thriving. There is not one single structure or physiology that will be optimum for every organism. No organism is a pinnacle of evolution. Botanists are working harder than ever trying to figure out how plants are related, which clades are valid. The following are the ones you should know as you continue into this book. Plants are a clade. True plants originated from green algae only once. The technical name for plants, to distinguish them from algae, is embryophytes, but this is used in only the most technical writing. It had been suspected that mosses, liverworts, and hornworts might have had a completely separate origin. Perhaps they did not descend from the same ancestor that produced all the other plants. But the sequences of nucleotides in the genes of bryophytes are so similar to those of all other plants that we are now confident they too are true plants. The terms vascular cryptogams and fern allies are handy but not accurate. Early diploid plants diverged into two clades, both with vascular tissue and without seeds. One clade, which we call the Lycophyte Clade, was very successful initially and evolved to be large trees with many complex features, but almost all became extinct. At present, the surviving descendants are Lycopodium, Selaginella, and a few other genera. These are the fern allies you will see in wildflower books. The second clade was a bit slower at the start, and for millions of years was overshadowed by the abundant members of the Lycophyte Clade. Many of the early members became extinct, but this clade produced all the ferns and seed plants. The term most often used now is the Euphilophite Clade. The ferns make up a subclade of the Euphilophytes, and they have been very successful without having seeds. Ferns thrive in all sorts of habitats, not only shady, damp forests, but also hot, dry deserts. They never produce wood, so they are all herbs, even the very tall ferns called tree ferns. Some ferns have evolved to have sophisticated relationships with ants, relationships that are mutually beneficial. The ferns make up a well-defined subclade within the Euphilophite clade, but we are not certain how to group the remaining vascular plants. Many are known only as fossils of isolated leaves or wood or reproductive organs. It is very rare to ever find a fossil of an entire plant with all parts still attached to each other. Some especially intriguing fossils look like fern leaves with seeds on them, so they were named seed ferns. But no seed fern is alive today, and no living fern ever has anything that even slightly resembles a seed. We believe that the name seed fern was a poor choice, but by now it is very commonly used. The many fossils of seed ferns are too incomplete for us to know if there was only one group of early seed plants or several. If there was only one group of seed ferns, then probably seeds evolved just once. If so, all seed plants make up one clade, the seed plants. But if seeds evolved 2 or 3 times, then seed plants is not a clade, not a closely related group that all descended from the same common ancestor. If seeds evolve several times, seed plants would then be the name of a grade and could only be used informally. Other extinct seed plants lived after seed ferns. Many look like intermediates in the evolution of modern seed plants from seed ferns, but the picture is not yet clear. Many people are taking a cautious approach now, tentatively considering all the seed plants as being related. They assumed that seed plants evolved only once, and then diverged into several major groups, most of which have some members that are still alive today. According to this theory, the early seed plants gave rise to these clades: cycads, cnidophytes, ginkgo, conifers, and angiosperms. Until recently, it was thought that all members of the angiosperm clade were either monocots or dicots. We used to think that after the first flowering plants originated, the group immediately diverged into 2 distinct clades, monocots and dicots. But new evidence indicates that split did not occur immediately. Early flowering plants evolved and diverged into several groups before the monocots originated and became so distinctive. The term dicot had referred to all the angiosperms except monocots, but like the term gymnosperm, this is unacceptable because it does not include all the descendants of the common ancestor of the group. At present, we use the terms basal angiosperms for those angiosperms that diverged early, monocots for the clade of lilies, palms, agaves, orchids, and their relatives, and eudicots for all the rest of the angiosperms. You will still see the term dicot used in many books, and it most often means basal angiosperms plus eudicots. Although the terms dicot, gymnosperm, and fern allies are being abandoned, it is good for you to know why they were invented and how they were being used. Even though you will still see them occasionally, it is best if you avoid using them. Figure 2:34. These avalanche lilies are shown as representative monocots, but the monocots are a large, diverse group. Many have large showy flowers, although flowers of grasses and sedges are small. Most monocots have parallel venation in their leaves, and many die back to a persistent subterranean bulb or rhizome. These Joshua trees are shown as another representative type of monocot. Many people think of monocots as small, delicate plants like lilies, irises, crocuses, and so on, but many, such as Joshua trees, become very large and tough. Other strong monocots are palms, agaves, yuccas, bamboos, and many bromeliads. This lupin, Lupinus latifolius, is shown as a representative eudicot, but eudicots are a gigantic and diverse group. Of the plants you see every day, unless a plant is obviously a monocot, conifer, or fern, then it is probably a eudicot. Eudicots can be trees, shrubs, herbs, vines, parasites, and succulents, as well as many other types of plants. Botany and beyond. Box 2-4 Noah's Flood and Population Biology. The biblical story of Noah's flood in Genesis provided a powerful stimulus for developing the sciences of population biology, biogeography, and evolution. Various biblical scholars had studied Old Testament texts, the Jewish Torah, and archaeological evidence, trying to establish dates such as the birth of Jesus and the construction of Solomon's temple. Although there is no archaeological evidence, various Bible scholars concluded that the flood of Noah occurred in 2348 BC. Noah's flood was of special interest to early scientists because at that point, according to the Bible, all life on earth had been destroyed by God except for the creatures in the ark. Scientists formulated the question: do plants and animals reproduce rapidly enough to repopulate a completely denuded earth in the time since Noah? Thus a scientist posing this question in 1700 AD, for example, could assume that Earth had been repopulated in 2348 plus 1700 is equal to 4048 years. First, consider the Bible passages that the scientists relied on. Genesis 7 verses 2 and 3 states that God told Noah to take 7 pairs of every kind of clean animal, only 1 pair of every kind of unclean animal, and also 7 pairs of every kind of bird. Genesis 7 verse 20 states that the flood completely covered the tops of all mountains. That means that there was no dry place anywhere that animals could use as a refuge and survive. Although we are all familiar with the rain lasting 40 days and 40 nights, the Bible states that it took a long time for the waters to drain away after the rain stopped. For the early scientists, the critical passages are Genesis 8 verse 4 and 5 that state that the ark was floating for 7 months before it settled on Mount Ararat, and it was 10 months before the tops of other mountains were visible and Noah opened the doors. Especially important was the explicit statement that God's plan succeeded. Genesis 7 verse 23 states that everything outside the ark had been killed. In the book of Genesis, God had set up the perfect experiment in population biology. All animals had been killed, and Earth had to be completely repopulated with all types of animals and birds, starting with just 1 male and 1 female of each of the unclean animals and just 6 males and 6 females of each of the clean animals. Could just 12 cattle breed rapidly enough to produce all the herds and wild cattle on Earth in 1700 AD? Could just 2 pigs be the source of all swine now present? The first step in answering this was to determine the fecundity of animals. How old do they have to be before they produce their first young? How many young do they produce in each litter? How many survive long enough to breed? And how many sets of offspring do they produce in their lifetime? Fortunately, it was about this time that wealthy landowners began keeping track of these sorts of numbers for cattle, poultry, fish, fowl, and even the deer in their private forests. The answer for every type of animal was the same: yes, all animals produce young rapidly enough to repopulate Earth in much less than 4,000 years. The early scientists then began to refine their thoughts and reformulate the question. At present, we believe in a balance of nature. Plants capture carbon and energy through photosynthesis, then the plants are consumed by herbivorous animals or by pathogenic fungi and bacteria. The herbivores are eaten by carnivores. If there are too many herbivores, they damage the plants too much, and some herbivores starve, which in turn affects carnivores. An outbreak of a pathogenic fungus that damages plants will cause various animals to starve, and so on. The balance between all these factors changes from year to year, so the relative numbers of organisms also change yearly. But prior to the analysis of Noah's flood, people believed that a different kind of balance of nature was maintained by God. God had created the various carnivores, herbivores, and plants such that plants would prosper just enough to supply the herbivores with exactly the right amount of food, and likewise the number of herbivores would be perfectly correct for the numbers of carnivores in the world. The balance would be perfect, never changing, and with no waste or shortage unless God specifically causes a drought or pestilence. As early scientists studied animal fecundity, they realized that the numbers did not fit together to make a divinely balanced system. All organisms are too fertile. If the previous interpretation of Genesis were correct, God had created plants and animals with the wrong properties to fit into a system that would be perfectly balanced and unchanging to the end of time. They realized that the story of Genesis as well as their own research pointed not to a stable, harmonious, garden-like Earth but rather to one of change and competition, of some organisms increasing at the expense of others. Population biology was born and has thrived ever since. Overview of plant ecology. Plants live in a complex, dynamic, and changing world. Earth's complexity is easy to see. Some areas are mountainous with cold temperatures and thin, rocky soil, but other areas are low, warm, and moist. Volcanoes release lava and ash that are initially sterile but that gradually change into rich acidic soil. In contrast, limestone breaks down to form alkaline soil. In addition, some soils are so rich in certain minerals they are toxic and plants cannot grow on them, whereas other soils have a balance of mineral nutrients that encourages luxuriant plant growth. On mountains and in valleys, the slopes that face the equator receive more sunlight and are much warmer than the sides that face away. Earth is dynamic, energetic, for many reasons. As Earth rotates on its axis, a particular area faces the hot sun during the day but faces the extreme cold of outer space at night. Throughout a year, seasons change from spring to summer and so on, each characterized by the amount of energy available from sunlight. Earth's atmosphere and its oceans are two gigantic fluids that circulate, moving energy from place to place. We experience the circulation as cold fronts of frigid polar air that move southward, pushing warm air aside, or as storms and hurricanes that carry water and energy in such concentrated forms they destroy buildings and push over trees. Earth's hot interior is another source of energy that affects life on the planet's surface. Erupting volcanoes catch our attention, but Earth's interior heat also powers continental drift. Hot, molten rock rises along the center line of each ocean then spreads outward as newly formed sea floor. This pushes the old sea floor to the side, which in turn pushes continents from place to place. North and South America formerly were widely separated by thousands of miles, but the continental drift of each changed direction and now they are moving toward each other. Central America is the result of this collision, which is still occurring. North and South America first made contact with each other less than 2 million years ago. As they continue to push into each other, they crumple Earth's crust, pushing Central America upward and causing numerous volcanic eruptions throughout the region. As the two continents finally came into direct contact, many animals and plants could migrate from one to the other. For example, armadillos have been slowly migrating from South America into North America, and have only recently arrived in the southern United States. Unfortunately, saber-toothed tigers migrated south, and as they spread into South America, they killed off many large animals, driving many species to extinction. Cacti originated in South America and were confined to just that continent until continental drift finally brought North and South America together. Cacti could migrate north into Mexico and the deserts of the United States as animals carried their seeds. The creosote bush that gives all the deserts of the southwestern United States their distinctive aroma also came from South America. Earth is a changing world. For comparison, think about our moon. It is complex but not dynamic. The craters we see today are the same as they were thousands or millions of years ago, with the only new activity to ever occur is an occasional meteor strike. But Earth is never the same, not even from one year to the next. Mountains are lifted by volcanism or continental collision, and they are worn down by erosion. Flooding rivers carry silt that settles into rich deltas and marshlands filled with life. Fires drastically alter the numbers of plants and animals in forests and prairies that burn. As continents drift, their changing positions affect the circulation of the oceans and thus the circulation of heat and moisture. During the time North and South America were widely separated, the Pacific and Atlantic Oceans were in contact and water circulated around each continent. But once the continents collided and formed Central America, oceanic circulation was altered. Water was forced to flow northward and southward, carrying warm water toward the poles and making those areas warmer than they had been before. Plants and animals located in the far northern parts of North America and Europe experienced climatic warming. Figure 2:35. This cactus, Stetsonia corine, is at home in Argentina. It is one of many South American cacti. For almost any cactus you would encounter north of the Panama Canal, there are similar but unrelated species in South America. There were no cacti in North America until continental drift brought North and South America close enough together for seeds to be carried northward. Figure 2:36. Creosote bush is at home in the deserts of North America, but its closest relative, Larica divaricata and two other species of Larica, live in South America. The plants in South America resemble those in North America so closely that we believe that at some very recent time, a seed or two of L. divaricata was carried northward the entire distance and then thrived in the new habitat. Once in North America, the plants gradually evolved just enough that we consider them to be a new species, L. tridentata, distinct from the ancestral L. divaricata. Much earlier than that, just before plants evolved from green algae, all continents had drifted together into one gigantic continent called Pangaea. This was so enormous that its center was a desert. Storms blowing in from the oceans would rain themselves out before they could reach the center of the supercontinent. But because all land was connected, the newly evolved plants could spread easily from place to place such that by the time Pangaea broke apart, each new continent carried some plants, and no continent was left without any plants. The first splitting of Pangaea ran from east to west, creating two gigantic continents, Laurasia and Gondwana. Imagine if Pangaea had broken apart before plants evolved from algae. Plants would have originated on one but not the other. On one continent, plants would have spread and diversified, and would have evolved into many clades. But the other continent would have remained barren until at some point millions of years later, some spores or seeds would have somehow blown or drifted to it. A crucial cause of Earth being a changing world is life itself. Living organisms take up energy and nutrients from soil, water, and air and then release their waste products back. Plants take so much carbon dioxide out of the air that they have changed the atmosphere. And because carbon dioxide is a greenhouse gas, the changing levels of carbon dioxide affect the temperature of Earth's surface, the biosphere, the place where life occurs. Photosynthesis produces oxygen as a waste product that is released into the air. Before the evolution of photosynthesis, Earth had no free oxygen at all. There was no aerobic respiration nor were there any animals. The photosynthetic production of oxygen is carried out by cyanobacteria, algae, and plants. Without these organisms, animals and fungi could not exist. Similarly, as animals and fungi respire their food, they give off carbon dioxide, which of course is valuable for plants. Thus photosynthetic organisms have changed Earth such that it is a better habitat for organisms that respire, and vice versa, organisms that respire are a source of the carbon dioxide needed by photosynthetic organisms. Figure 2:37. For most of us, it is difficult to imagine what Earth was like before the evolution of true plants. There was no life on land at all. For billions of years, life existed as bacteria and archaeans in the oceans. Later there were fish and algae, but nothing at all on land until at the earliest, about 450 million years ago. Most of the surface was rock, and if any soil particles formed, they would have mostly been washed into the oceans because there were no plant roots to hold them in place. The area shown here is in Death Valley National Park, which does have plant and animal life, but it is widely scattered and confined to areas with at least occasional water. Plants, animals, and all other organisms interact in more immediate, direct ways as well. Interactions may involve one organism benefiting by harming the other, by both organisms helping each other, by one organism benefiting without harming the other, and by one organism harming another with no benefit to itself. Herbivores and omnivores are examples of predation. They benefit by eating plants, and this of course harms the plants, so plants need to defend themselves from animals. There is no one single optimal defense. Instead, many alternative types of defense have evolved, such as having spines or being poisonous. Pathogenic fungi and bacteria also prey on plants. Figure 2:38. The grass and herbs here capture energy and minerals, converting them into the organic molecules of the plants' bodies. Herbivores such as deer eat the plants, using the organic compounds as their own food. The respiration of the animals returns carbon dioxide back to the air. The animal dung also is broken down by fungi, releasing carbon dioxide. Thus there is a cycling of carbon dioxide, minerals, oxygen, and other components. In addition to large mammals such as deer, insects such as this caterpillar also consume immense amounts of plant material and are an important component of the carbon, nitrogen, and mineral cycles. Table 2:6. Direct interactions among organisms. Animals often harm plants without benefiting from the damage they do. This is an amensal relationship. For example, they trample plants as they walk across them or when they lie down. Here too it is beneficial for plants to somehow defend themselves. The presence of trees often creates a shady, cool area in which smaller plants, such as mosses and ferns, can live. The shade-loving plants benefit without affecting the trees. This is a commensal relationship. In mutualistic relationships, both organisms benefit. When animals pollinate flowers, both the plant and the animal benefit. The animal obtains nectar or pollen as food, and plants have their pollen grains carried from one flower to others, improving cross-pollination and sexual reproduction. The evolution of fleshy, nutritious fruits is similar. Animals benefit by eating the fruits, whereas plants benefit from having their seeds dispersed, often in a pile of organic fertilizer. These cases of mutualism differ fundamentally from those of predation and amensalism. In predation, for example, the organisms are battling against each other. When an animal eats a plant, some surviving seedling of that plant might have a defensive mutation that might make it more spiny or poisonous than its parent was. If so, that seedling is more likely to survive and, over many generations, spininess or toxicity may gradually increase in that species of plant. But if so, then it will be helpful to the animals if they have mutations that allow them to overcome the plant's new defenses. This battle between plants and animals has continued for the hundreds of millions of years that both have existed. Figure 2:39. This pasture is being overgrown by prickly poppy because cattle and deer will not eat it even if starving. The leaves and stems are covered with spines. It contains a toxic milky juice as well. If there were no herbivores, the grasses might grow well enough to compete against the prickly poppy and drive it out, but with cattle eating the grass, the prickly poppies have a distinct advantage and might eventually take over the entire pasture. Figure 2:42. Trees have died and fallen across the stream, which keeps the road moist enough for moss plants to grow on them. The mosses are benefiting from the presence of these dead trees, but the trees are deriving no benefit at all from the mosses. Similarly, surrounding trees that are still alive provide shade that is beneficial to the mosses, but the mosses provide no benefit to the living trees either. Figure 2:41. This pile of Texas lignum vitae seeds has passed through an animal, probably a javelina, so all the fruit pulp has been digested but the seeds are intact. 5 have already germinated. The javelina probably ate the fruits far from this area and then defecated here, thus spreading the seeds away from the parent plant. Both the plant and the animal benefit in this mutualistic relationship. But a completely new type of evolution began to occur as animal-pollinated flowers evolved. Because plants benefit when an animal carries its pollen to another flower of the same species, any mutation in the plant that helps the pollinator will be beneficial to both. Similarly, any mutation in the animal that makes it a better pollinator helps both itself and the plant. This is called coevolution, and it causes certain plants and animals to become well adapted to each other. Keep in mind that at the same time that the plant's flowers are producing nectar that benefits a pollinator, the plant's leaves may be producing poisons that keep herbivores away. Plants interact with each other. Of course, plants never eat each other. Instead, plants compete with each other for water and minerals in the soil, for sunlight, and for the attention of pollinators and seed dispersers. In an oak forest, for example, trees may grow so close together that the roots of any one tree touch those of the surrounding trees and there is no room for a new oak seedling. Because all the trees are the same species of oak, they probably are almost identical genetically and each is about as good as any other at absorbing water and nutrients. Oaks are wind-pollinated, so they are not competing for pollinators. But as mentioned before, there are many ways of being adapted, so there might be small wildflowers growing between the oaks, wildflowers that can tolerate growing in shade and whose small root systems will fit into any spaces between the roots of the oaks. Habitats are complex, so there may be rocky cliffs that are too dry for the oak trees but are just fine for junipers. In this forest, the oak trees compete with each other for water, minerals, and light. They make the habitat shady enough for wildflowers, and the junipers are using a resource that oaks cannot. Figure 2:42. Orchids have extremely complex flowers as the result of coevolution between each species of orchid and one particular species of pollinator. The benefit of this to the plant is that a pollinator carries the orchid's pollen only to other flowers of the same species. None is wasted by being accidentally carried to a different species of plant. On the other hand, if the pollinators die due to a disease or bad weather, the orchid then has no alternative means of transferring its pollen. This phalaenopsis is popular in cultivation. Because all organisms evolve, a set of organisms such as a forest never stays the same. At the same time, the physical environment also is changing, due both to geological processes and the effects of organisms. The biosphere is complex, dynamic, and changing. The plants, animals, geology, and climate that were present in the past were different than they are now, and those of the future will also be different. But for studying and understanding life, the process, principles, and concepts are always the same. At the next level. 1. Alternatives and consequences. A theme of this book is to ask what are the alternatives, and what are the consequences of various alternatives. Parasitic plants are discussed at several points because they often represent extremely unusual alternatives to the biology of non-parasitic plants. I recommend the book Parasitic Flowering Plants by Henning S. Heidi Jorgensen because it provides many interesting examples of highly modified plants and is well illustrated. Even as a beginning botanist, you will understand most of it and benefit from knowing how extreme certain alternative can be. You might find it difficult to believe that some of these are actually plants. 2. Evolution by natural selection. Charles Darwin and Alfred Russel Wallace discovered evolution by natural selection in the 1850s, and both evolution and natural selection are dominant themes in this textbook. Darwin wrote The Origin of Species in 1859, and it was the crucial introduction of basic principles to the world. However, I recommend at this point that you read Darwin's Voyage of the Beagle, 1989, Penguin Books, which is his account of his trip and his collecting. It is written as a narrative and is wonderfully easy to read. Darwin wrote it shortly after he returned from his voyage, many years before he had figured out the principles of evolution by natural selection. As you read Voyage of the Beagle, you will have a better understanding of evolution than Darwin did when he was writing it. In certain parts of the book, it is fascinating to see how Darwin puts the clues together correctly and comes to key understandings using the scientific method. But in other parts, it is excruciating to read as he misses the clues and misses the point. Voyage of the Beagle is an excellent example of how the scientific method is used, how Darwin's thought processes and reasoning finally allowed him to see the principle that is the basis of all aspects of biology. Summary. 1. Almost all plants consist of stems, leaves, and roots. Flowers and cones are modified stems and leaves. 2. One of the defining characters of angiosperms is that their seeds always develop inside of fruit. 3. The internal organization of plants is very uniform and simple. Virtually all stems have epidermis, cortex, vascular tissues, and pith. Roots lack pith. Leaves consist of epidermis, mesophyll, and vascular tissues. 4. All plants have a primary body, but only woody plants have a secondary body, which consists of wood and bark. 5. All organisms must take in energy and raw materials from their environment. They also give off waste products to the environment. 6. All known organisms store heritable information in DNA. 7. Plants detect and respond to information from the environment as well as from various organs within the plant itself. 8. A grade is a level of evolution, and it is an artificial group because various unrelated organisms might evolve to a particular level independently. A clade is a natural group because it contains a common ancestor and all the descendants of that ancestor. 9. There are multiple ways for an organism to be adapted to its environment. There is no single optimal solution to any biological problem. 10. The biosphere is constantly changing due to physical and geological forces, as well as to the presence of life itself. 11. Organisms interact with each other, often with animals and fungi consuming plants and plants needing to defend themselves. But some interactions, such as pollination, are mutually beneficial to both plants and animals. Plants also compete with each other for various resources. Important terms: Angiosperm, basal angiosperm, Biosphere clade, common ancestor Cormophyte, cryptogam dicot, embryophyte eudicot, Euphilophite Clade, Fern, Gondwana grade, Gymnosperm, herb, Laurasia, Lycophyte Clade, monocot, non-vascular plant, Pangea, phloem, photosynthesis plant, reproductive organ, respiration, seed plant, spermatophyte, spore-bearing plant, thallophyte, thallus, tracheophyte, vascular plant, vegetative organ, woody plant, xylem. Review questions. 1. Name the 3 vegetative organs of a plant. 2. How do the reproductive organs of ferns differ from foliage leaves? 3. Name the 4 basic organs of a flower. Describe situations in which flowers might not need 1 or 2 of these organs. 4. Name the 2 vascular tissues found in vascular plants. What does each transport? 5. Are there any types of organisms that do not need to take in energy and raw materials from their environment? Discuss the source of energy for plants as opposed to animals. Describe some of the raw materials that are needed by plants. 6. Is the central metabolism by which plants acquire energy. It occurs only when and are present. 7. Each plant receives a great deal of information from its parents by means of the DNA in which 2 cells. Describe the types of information that plants receive from the environment. Why is it that environmental information cannot be stored in DNA? 8. The activity of the roots of a plant must be coordinated with the activity of leaves. Describe how this occurs. What type of information molecule is used? 9. Which 3 groups of plants are referred to as bryophytes? They differ from all other plants in 2 main features. Name the features. 10. Which group of plants does the term angiosperm refer to? What are the 3 main subgroups of angiosperms? Do angiosperms have vascular tissue? Do they have seeds? In conifers, seeds develop inside cones, but in angiosperms, seeds always develop inside of. 11. Evolution by natural selection causes organisms to become more adapted to their environment. Is the environment uniform across all of Earth? Does the environment change with time? Considering this, do you think that there is only one single optimum way for a plant to be adapted? If not, what is the alternative? 12. Have North and South America always been connected by Central America? Once the 2 continents joined, plants and animals could move from one to the other. Animals can walk, fly, or swim, but how does a plant species spread from one area to another? South America has thousands of plants adapted to the Amazon rainforest. Do you think it is likely that any of those plants will be able to migrate to North America across the deserts of Mexico, Texas, and Arizona? 13. The concept of biosphere was touched on only briefly in this chapter, but you have many resources in this book and online to explore this concept. What are some of the geological and biological components of the biosphere? We humans are burning fossil fuels and adding carbon dioxide to the atmosphere. How does this affect the biosphere? 14. Plants and animals usually battle each other. Describe why and how this occurs. Once animal-pollinated flowers evolved, a new type of plant-animal interaction was possible. Describe it. How is it possible for a mutation in a plant to be beneficial for both the plant and for an animal? 15. Earth is constantly changing, and so are all organisms as they evolve. But despite so much change, it is possible to study and understand life because several things are always constant. What are those things? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated, Natalia Ora, purple tulip, Shutterstock Incorporated Marie C. Fields, dandelion, Shutterstock Incorporated Daniel Chrysal, poppy, Shutterstock Incorporated Saryuri, plant icon, Shutterstock Incorporated Vector, Digging Man icon, Shutterstock Incorporated ZArt. Part 1. Plant Structure. Chapter 3 Cell Structure. Chapter 4 Growth and Division of the Cell. Chapter 5 Tissues and the Primary Growth of Stems. Chapter 6 Leaves. Chapter 7 Roots. Chapter 8 Structure of Woody Plants. Chapter 9 Flowers and Reproduction. Part Opener Image. The structure of this poppy fruit is adapted such that it aids the survival of the plant. While immature, the fruit protected and nourished the seeds as they developed. Now that the seeds are mature and must be released, pores have formed near the top of the fruit. During the formation of the pores, some cells broke down, creating flaps, then cells at the base of each flap contracted, pulling the flaps outward and downward, opening the pores. As the fruit sways in the wind, seeds will be thrown out through the pores in various directions. Plant structure is the physical, material body of a plant, composed of carbohydrates, proteins, lipids, minerals, water, and other components. In terms of both material and energy, it is expensive for an organism to build a body. All of the energy and resources used by a tree to construct its body could have been used instead for reproduction, increasing its number of offspring. The theory of evolution by natural selection predicts that organisms must receive enough benefit from their structure to offset the cost of building it. It must be more advantageous selectively to have a body than not to have one. The benefit is that the structure is the framework in which metabolism occurs and the means by which metabolism interacts with the environment. This can be more easily understood by considering the alternative, organisms with very little structure. Several organisms have almost no structure whatsoever: viroids, viruses, and mycoplasmas. All are parasites that live, grow, develop, and reproduce only if immersed within the body of a host organism. The host provides a benign environment with enough nutrients for the parasite while also being free of harmful chemicals. The temperature of the host's body is ideal for the parasite, and adequate water is available. Parasites cannot live outside the host-supplied environment. With minimal structure, they have minimal capacity to interact well with the heat, cold, drought, and scarcity of nutrients in a natural environment. Organisms whose bodies are more structured and complex are able to resist temporary adverse environmental conditions as well as exploit optimal ones. A simple bacterium can absorb mineral nutrients from certain environments, but, by constructing a root system, a plant can spread its mineral-absorbing metabolism throughout an extensive, deep volume, growing past dry, rocky, mineral-poor soil and tapping moist, rich, fertile soil. The investment for the plant is great, but sufficient reward is achieved in having an adequate, secure source of water and minerals. The structures discussed in this part of the text are important to the plants themselves, but they are also important to us because we rely on them for food and many products, and as we cultivate them we affect the lives of many other organisms. Most of the food we eat is some part of a plant body. For example, we eat the leaves of lettuce, spinach, and cabbage, roots of carrots, beets, and radishes, stems of asparagus and potatoes, and fruits and seeds such as apples, oranges, peas, beans, and almonds. Some of our foods are so highly processed we may not immediately realize they were once part of a plant body. Bread, donuts, cakes, and pasta are made with flour, beer is brewed from barley and hops, and wine is fermented grape juice. Our very lives depend on these plant-based foods because they supply us with energy, vitamins, and minerals. Look at your hand. Every atom and molecule, every bit of your body was at one time part of some plant. Vanilla, chocolate, cinnamon, sage, black pepper, and chili peppers are spices that do not really provide us much in the way of nutrition, but they make eating fun and few people would want to give them up. The cotton in our clothes is a plant structure, as are the fibers in paper and burlap bags and the lumber used for construction or for making guitars and pianos. Our reliance on plant bodies goes beyond these fundamental needs. The grass lawns on which we play or relax on are the leaves and stems of plants, and trees provide us with shade and protection from weather. We love the beauty of flowers, and almost certainly there will be some point in your life when something wonderful or tragic will have happened and you just won't know what to say. At that point you will probably give someone flowers to express emotions you cannot put into words. We celebrate weddings and birthdays with flowers, we give flowers to friends when they are ill or just for friendship, and we rely on flowers to express sympathy at funerals. Because we rely on plants for so many things, we need to cultivate them on farms, orchards, and tree plantations, and this raises important issues with the rest of the world. Agriculture began as early as 9,000 years ago and has been affecting the world ever since. The first step of agriculture then as now is to clear a piece of land of its natural vegetation and animals. If we apply fertilizers or pesticides to crops, rain may wash these chemicals from the fields into nearby streams, polluting them and harming fish, frogs, and other aquatic life. Crops must be harvested, processed, and transported to markets, all of which require roads, fuel, and machinery and result in pollution. Our use of plant structures for so many things in our lives results in negative impacts on the ecology and well-being of many organisms. Plant structures, however, may also provide solutions to these problems. Scientists are conducting research to find plants that provide more abundant or more nutritious food, better fibers, or natural resistance to insect pests. Many of our crop plants now produce much greater harvests than did the crops available just a few decades ago. For example, a fast-growing plant called kanaf can produce many more tons of fibers per acre than do pine trees, and thus less land is needed to make the same amount of paper. It may even be possible to convert some farmland back to a more natural conditions suitable for wildlife. Most plants around us are angiosperms, commonly called the flowering plants. Most plants discussed in this text are angiosperms, and to a lesser extent we discuss ferns and conifers. One of the most noteworthy features of angiosperms is the diversity of bodies that occur in this group. Some angiosperms are ordinary herbs or shrubs or trees, but some are desert succulents, vines, parasites, bulbs, and tubers. Ferns and conifers are not so diverse, and this text would have been much shorter if it had been written 135 million years ago before angiosperms had come into existence. Angiosperms are one of the newest groups of plants. The first true plants originated about 420 million years ago, but the oldest fossils of angiosperms are only about 135 million years old. At the time angiosperms originated, the world was a very different place. The air and land were much warmer, as were the oceans. Consequently, much more water evaporated from the oceans into the air and then fell as rain on land. Warm, moist conditions allowed the growth of lush vegetation that consisted mostly of ferns and conifers. The dominant land animals were dinosaurs. During the last 100 million years, Earth warmed even more as volcanoes and lava released large amounts of the greenhouse gas carbon dioxide into the air. However, gradually plants took carbon dioxide out of the air as they used it in photosynthesis and built their bodies with it. As plants removed carbon dioxide from the air, less heat was trapped and Earth began to cool. Starting about 35 million years ago, it entered the Pleistocene Ice Age 2.5 million years ago. It was during this time of changing temperatures and climates that the angiosperms originated and evolved to have such diverse bodies. Continental drift is another factor that affected the evolution and diversification of the flowering plants and the structures that make up their bodies. Continents are constantly changing their position on Earth's surface as hot, fluid rock flows in Earth's mantle below its crust. As angiosperms were diversifying, North America broke away from Europe and drifted westward, whereas South America separated from Africa and Antarctica and migrated west and northward. Today, North and South America are colliding, pushing up a range of mountains we call Central America. As the continents change their position, they move from warm areas near the equator to cold areas near the poles or vice versa, and they block circulation of ocean currents. These currents contain millions of tons of water that is either warm if it is flowing from the equator, or cold if coming from the poles. As continents shift, they redirect the flow of that water and that heat. For example, Alaska is not as cold as it would be if warm water did not flow from the equator, past Japan and up to Alaska's southern coast. Thus, the changing climate of Earth and the changing position of the continents created an extremely varied and variable environments to which plants had to become adapted. Those that did not adapt became extinct. As you study the material in this part, think about how the structures discussed facilitate the metabolism that occurs inside them. How do the structures make the metabolism more efficient? What are the selective advantages of the structures? Are alternative structures possible for a particular metabolism? If so, what are the consequences of each? The structures that make up the body of a plant must work together to make a functional plant. And they must evolve. They evolved over long periods of times and in conditions that were very different from those we live in now. If the past climate of Earth had been different, the structures covered in this text would also have been different. Chapter 3 Cell Structure. Chapter Opener Image. Several types of cells are present in this cross-section of an anther in a lily flower. In the center of each chamber are pollen grains, cells that carry genetic information during a plant's reproduction. Each chamber is lined with a layer of tall cells that help pollen grains mature, and the large cells just below the anther's surface will break the anther open such that the pollen grains can be carried to another flower. Each cell type has its own characteristics and modifications, and all types must work together. Outline. Concepts. Membranes. Composition of membranes. Properties of membranes. Basic cell types. Plant cells. Protoplasm. Plasma membrane. Nucleus. Central vacuole. Cytoplasm. Mitochondria. Plastids. Ribosomes. Endoplasmic reticulum. Dictyosomes. Microbodies. Cytosol. Microtubules. Microfilaments. Storage products. Cell wall. Fungal cells. Associations of cells. Box 3 to 1 alternatives, unusual cells. Box 3 to 2 plants do things differently, calcium, strong bones, strong teeth, but not strong plants. Box 3 to 3 botany and beyond, the metric system and geometric aspects of cells. Learning objectives. After reading this chapter, students will be able to describe the benefits and negative consequences of unicellular and multicellular organization. Summarize the composition of membranes. List the properties of membranes. Name the 2 basic cell types. Recall and define the 15 organelles found in plants. Identify the components of plant cell walls. Describe 2 communication methods of multicellular organisms. Did you know? All organisms are composed of cells. The bodies of some algae consist of just a single cell, but all plants have multicellular bodies. Giant trees contain trillions of cells. Many cells must be alive to be functional, whereas others must be dead. Some plant cells are large enough to be seen with the naked eye. Examples are the finest strands composing cotton thread and the filaments along the torn edge of paper. Concepts. All organisms are composed of small structures called cells. In plants, each cell consists of a box-like cell wall surrounding a mass of protoplasm, which in turn contains its own smaller parts, the organelles, such as nuclei, mitochondria, and chloroplasts. Cells are also the physical framework within which a plant's metabolism occurs. Water and salts are absorbed from soil by root cells, they are transported throughout the plant by cells of the vascular tissues, and the energy of sunlight is used in leaf cells to convert carbon dioxide and water to carbohydrates. Plant reproduction is also based on cells and cell biology. Some cells in flowers produce pigments or nectar that attracts insects that carry pollen between flowers, allowing sperm cells to contact egg cells. Figure 3, one light micrograph showing several basic features of plant cells. The walls and chloroplasts are easily visible. Other organelles occur but are difficult or impossible to see by light microscopy. Figure 3.2 Plant metabolism, development, and survival depend on numerous cells working together in a coordinated, integrated fashion. These cells store water in the center of a sunflower stem. They are relatively large and filled mostly with water. The cell walls and a nucleus are visible, part of the system that conducts water and nutrients in a sunflower stem. Numerous types of cells occur in specific arrangements that permit efficient conduction. The large red cells in the center conduct water, the small gray cells above them conduct sugars. The cells at the top have thick red walls and provide strength to the stem. In this transverse section through a leaf of ligustrum, you can see a variety of cells. Those in the center carry out photosynthesis. Note, the micrograph in Figure 3.2b is 60 mm tall and has a magnification of 60. If we were to shrink the micrograph down to life size, the micrograph would be 1 mm tall and you would be able to see some of the cells with a magnifying glass. Figure 3.3 A micrograph made with a transmission electron microscope of leaf cells. Especially important are the numerous membranes. Table 3.1 Examples of plant cell shapes and sizes. Considering the large number of living organisms and the numerous types of metabolism that must be carried out, one might suspect that there are hundreds of types of cells, but actually, just a small number of cell types exist. Most differences between organisms are due to differences in associations of their cells, not in the cells themselves. Regardless of whether a root, stem, leaf, or flower is being constructed, the same basic units, cells, are required. Only the cell associations and minor modifications of the cells themselves change from tissue to tissue or organ to organ. Although only a few types of cells exist, their differences are important. Any organism composed of more than one cell always has several types, each specialized for different tasks. As a plant develops, the cells in various parts become especially adapted for specific tasks. This division of labor allows the entire organism to become more efficient. Unicellular organization has a significant consequence: it does not allow division of labor or specialization. Each cell must perform all tasks: sensing the environment, gathering nutrients, excreting wastes, defense, movement, and reproduction. Because each cell must perform all tasks, it cannot do any one very well. Mutations that make a cell well adapted for protection make it less adapted for other functions and therefore are selectively disadvantageous. The same is true for modifications that improve photosynthesis, reproduction, and so on. Figure 3.4 Transverse section of wood showing several cell types. Fibers with thick walls provide strength, and large open cells conduct water. These cells act as building blocks, and more wood is easily constructed by adding more cells. Stronger wood is produced by adding more fiber cells. More conductive wood results from adding more open cells. Table 3.2 Examples of plant cell types, specializations, and division of labor. Multicellularity and division of labor result in a more efficient organism, but they have negative consequences as well. As each cell becomes more specialized, it depends more on the others. If a cell evolves toward having thick walls and offering maximum protection, it must rely on other cells of the organism for photosynthesis, mineral absorption, and reproduction. Damage to one part of the organism may result in the death of all cells, even those not initially damaged, whereas in a unicellular organism, a cell dies only if it is damaged directly. Which is more advantageous selectively, a unicellular organism composed of one generalized cell or a multicellular organism composed of specialized ones? The answer is not so obvious. Both types have existed for hundreds of millions of years, and thus, both must be considered highly adaptive and successful. Also, selective advantage depends on environment. In certain habitats, unicellular organisms are better adapted, in others, multicellular organisms survive better. Like whole individuals, cells have a lifespan. During their life cycle, cell size, shape, and metabolic activities can change dramatically. A cell is born as a twin when its mother cell divides, producing two daughter cells. Each daughter cell is smaller than the mother cell, and except for unusual cases, each grows until it becomes as large as the mother cell was. During this time, the cell absorbs water, sugars, amino acids, and other nutrients and assembles them into new, living protoplasm. After the cell has grown to the proper size, its metabolism shifts as it either prepares to divide or matures and differentiates into a specialized cell. Figure 3, 5 cells located at the growing tip of pinus are specialized for cell growth and division. Their thin cell walls allow sugar, water, and other nutrients to enter the cells easily. However, these cells are too soft and weak to be useful as bark or the shell of a nut. These stone cells provide strength in a coconut shell and therefore protect the seed. Almost the entire cell volume is wall. Protoplasm was present when the cells were young, growing, and synthesizing their walls. However, after the walls were completed, cell metabolism was not necessary, and the protoplasts died. Such modifications make it impossible for a stone cell to be a dividing, growing cell as in. Both growth and development require a complex and dynamic set of interactions involving all cell parts. That cell metabolism and structure should be complex would not be surprising, but actually, they are rather simple and logical. Even the most complex cell has only a small number of parts, each responsible for a discrete, well-defined aspect of cell life. Membranes. All cells contain at least some membranes, and cells of eukaryotes contain numerous organelles composed of membranes. Membranes perform many important tasks in cell metabolism. They regulate the passage of molecules into and out of cells and organelles. They divide the cell into numerous compartments, each with its own specialized metabolism, and they act as surfaces that hold enzymes. Without membranes, life would be impossible. Indeed, many things that cause death in both humans and plants, heat, cold, many poisons, alcohol, kill because they disrupt membranes. Composition of membranes. All biological membranes are composed of proteins and two layers of phospholipid molecules. If phospholipids are poured carefully onto the surface of calm water, they automatically form a single layer on the water's surface, with their hydrophilic ends forming hydrogen bonds to water molecules and their hydrophobic fatty acids projecting out of the water. If the water is agitated, the lipid layer doubles over and makes a bilayer in which all fatty acids are away from water and all phosphate groups are in full contact with it. Any break or tear in a bilayer membrane exposes hydrophobic fatty acids to water, and thus, membranes always reseal themselves after a rupture. Figure 3.6 in electron micrographs, membranes appear as two dark lines separated by a light region. Pictures such as led scientists to believe that membranes consist of two layers of lipids with proteins located only on the surface. However, many proteins fit partly into the lipid bilayer or pass all the way through it. A phospholipid has three parts, two fatty acids, a backbone of glycerol, and a phosphate group. The lipid bilayer is a very thin solution. If it contains several types of lipid, they diffuse laterally throughout the membrane because they are not bonded to each other, but they cannot diffuse vertically from the membrane into the surrounding solution. All biological membranes contain proteins as well as lipids, usually in a ratio of 60% proteins and 40% lipid. Most proteins have large hydrophilic regions, and thus, they associate mostly with the phospholipid phosphates and with water. Many also have large hydrophobic regions that allow them to sink into the membrane and associate with the fatty acids. Variations in hydrophobic and hydrophilic regions mean that various proteins sit entirely on the membrane surface, are partly immersed in it, or span it entirely, with either end projecting out of opposite sides. This allows the protein to have its active site on either or both sides of the membrane or within it. Likewise, a protein may act as a hydrophilic channel that permits small hydrophilic molecules to pass through the membrane. Proteins that are even partially immersed in the lipid bilayer are said to be intrinsic proteins. Others, extrinsic proteins, are located outside the membrane and merely lie next to it. Figure 3.7 In quiet water, phospholipids form a monolayer at the surface. By projecting away from the water, the fatty acids do not disrupt the hydrogen bonding between water molecules. With agitation, phospholipids form a bilayer. This arrangement allows the greatest number of hydrogen bonds to form between phosphates and water while simultaneously minimizing the disruption of water-water hydrogen bonds by the fatty acids. Figure 3.8 Intrinsic membrane proteins differ in the size, shape, and location of hydrophobic, lipid-soluble regions. Consequently, some sink deep into the membrane, others span it, and some penetrate only one lipid layer. Some intrinsic proteins contribute to the membrane's fluid nature and, like the lipids, can diffuse laterally. Other proteins interact with adjacent proteins, forming complexes or domains different from surrounding regions of the membrane. If all membrane components could freely diffuse laterally, the membrane would become homogeneous and no differentiation could occur. However, because at least some proteins are bound to their neighbors, membranes are heterogeneous and patchy, and differentiation does take place. Because the membrane is a heterogeneous liquid, it is said to be a fluid mosaic membrane. Some membranes contain a small amount of sugar, usually less than 8%. The sugars occur as short-chain oligosaccharides, each with about 4 to 15 sugar residues. These oligosaccharides are bound to certain intrinsic proteins, converting them into glycoproteins. Rarely, sugars are attached to membrane lipids. Currently, we believe these polysaccharides make the membrane more distinctive and easy to recognize, an especially important feature in animals that have an immune system. Specialized protective cells attack and destroy anything that they do not recognize as a part of the animal's own body. The glycoproteins and glycolipids occur almost exclusively on the outer surface of the membrane that covers the cell, a position that allows maximum exposure for these polysaccharides. Glycoproteins and glycolipids may be less important in plants. Alternatives. Box 3-1 Unusual cells. Many plant cells are like those described in this chapter, but there are numerous types of unusual cells. Each example described here demonstrates an alternative way of organizing a cell, and of course, we can ask, what are the consequences of each alternative? Dead cells. Many plant cells die as a necessary part of their development. If they remained alive, they could not function as well, if at all. Cork cells in bark deposit a waterproof, enzyme-resistant chemical called suberin in their cell walls, and then they die. Suberin keeps water in the plant and resists attack by microbes, but what is the consequence of dying? By breaking down their protoplasm, cork cells become much less nutritious than if they remained alive. It does an animal no good to eat cork because there are no proteins, starches, lipids, nucleic acids, vitamins, or anything else of value. An animal would waste its energy chewing and trying to digest cork. Our own epidermis cells too are dead and are more protective than they would be if they were alive and nutritious. Water-conducing cells in plants, called tracheids and vessel elements, grow to their proper size and shape then undergo programmed cell death. When fully mature, each cell consists of just a wall and an empty space where protoplasm had been. Water is pulled upward through these empty cells, and if protoplasm were still present, it would impede water movement. Water-conducting cells cannot function while living, they must be dead. Other cells that function after dying are those of many plant hairs, shells around seeds, and thorns and spines. Unusual nuclei. Cells that develop into pollen grains in flowers are surrounded by a layer of cells called the tapetum. In many species, the nucleus of each tapetum cell divides once or twice more than the cells do, so at maturity, each cell has 2 or 4 nuclei. The consequence of this seems to be that each cell then has more capacity to control a very active, dynamic cytoplasm. In contrast, cells that transport sugars throughout a plant's body destroy their nuclei during development. Unlike water-conducting cells, sugar-conducting cells cannot conduct if they are not alive. What, however, is the consequence of destroying the nucleus? We do not know why this is necessary, but we do know they become reliant on adjacent cells to control their metabolism. Flagella. We think of plants as stationary and animals as mobile, but sperm cells of plants such as mosses, ferns, and even some seed plants have flagella and swim. If sperm cells lacked flagella, they could not get to egg cells. There is, however, another alternative in most seed plants. Their sperm cells lack flagella but are carried passively to egg cells by elongate cells called pollen tubes, each of which grows out of a pollen grain giant cells. Plant cells tend to be larger than animal cells, but both types are usually microscopic. Fortunately, several types of giant plant cells are extremely common and are so large you can see them with the naked eye. Cotton cloth and thread are made of long, slender cells, each up to 4 centimeters long. You can see individual cells by teasing cotton thread apart. These long cells grow from the surface of cotton seeds and then die and protect the seeds by forming a fluffy, soft layer. Most paper is made from water-conducting tracheids of wood, each tracheid being several millimeters long. These cells are easy to see by simply moistening a piece of paper and pulling it in two. The tiny filaments protruding from each edge are single cells. For these cells, a beneficial consequence of their extraordinary length is that it is easier to pull water through a few long cells as opposed to many short cells. Very unusual giant cells occur in some of the plants that ooze milky white liquid when poked in spurges in Euphorbiaceae. The milky sap is always at least mildly toxic, often extremely toxic, and is produced by cells called laticiphers. If quite a bit of liquid drips out, it is probably produced by long, tubular cells. Tubular laticiphers begin development as ordinary, small cells, but they continue to grow even after surrounding cells have reached their full size and stop enlarging. The laticiphers elongate, pushing into spaces between other cells, becoming ever longer, and often branching and growing in multiple directions. Unlike cotton cells and tracheids, these must remain alive at maturity, and have hundreds of nuclei directing the synthesis of milky sap. One consequence of being giant, branched tubular cells is that even if an animal chews into just one small part of a plant, toxic sap from a wide area of the plant will flow to that site and deter the animal. Unusual cells are mentioned here to show that even though most plant cells are rather similar to each other, there are alternatives, and with the alternatives come consequences. Most of this book focuses on the most typical, most ordinary cells, tissues, and metabolisms, but always think about the possibility of alternatives and how those might affect the biology of the plant. Figure B3, 1. The long, branched tubes here are laticiphers, each has branched many times, and all tips elongate and intrude between other cells. Individual cells are very long and have hundreds of nuclei. Properties of membranes. Membranes have several important properties. First, they can grow. Membranes are formed molecule by molecule in certain regions of the cell, then entire pieces of membrane are moved as small bubbles or vesicles to different sites in the cell. When the vesicle of preformed membrane arrives at the growing membrane, the two fuse. In addition to permitting movement of membrane pieces, membrane fusion allows the transport of material. The volume inside the vesicle may be filled with substances that must be accumulated, broken down, or otherwise metabolized at the vesicle's destination. Vesicle movement may also release material to the outside of the cell. This exocytosis is a means to excrete almost anything: wastes, debris, mucilage, proteins, and polysaccharides. For example, roots slide through the soil by secreting a slippery, lubricating mucilage formed within root cells, packaged into vesicles that migrate to and fuse with the cell's surface membrane. This mucilage is then released to the exterior. In many flowers, nectar is secreted from glands by exocytosis. Endocytosis is basically the opposite process. A small invagination forms in the outer membrane, then pinches shut, creating a new vesicle that contains extracellular material. Endocytosis is especially common in algae and other microscopic organisms that take in food particles this way, but it is not known how common it is in plants. Permeability is an important property of membranes. All biological membranes are selectively permeable membranes, meaning that certain substances cross the membrane more easily and rapidly than others. Because large regions of a membrane are mostly lipid, membranes are more permeable to hydrophobic substances than to anything that carries an electric charge. However, if charged compounds such as inorganic salts, sugars, and amino acids could not enter cells at all, or if they could enter only slowly, cells would starve. Movement of charged substances is assisted by large intrinsic proteins that span the membrane and act as hydrophilic channels through it. This has facilitated diffusion. Other proteins, called molecular pumps, actually bind to a molecule on one side of the membrane, and then by using energy, the protein changes shape and releases the molecule on the other side. By using this active pumping, called active transport, cells accumulate substances until the interior concentration of solute far exceeds the exterior concentration. A pump working in the opposite way can actively transport materials out of the cell. Except for the pumps, the membrane must be impermeable to the molecule. It would do no good to have a molecular pump for a type of molecule that could easily leak back through. All life depends on the principle of compartmentalization, the formation of many compartments, each specialized for a particular process, such as producing a particular substance or using a particular precursor. For example, our blood is compartmentalized away from our digestive system. The two must work together, but they must not be mixed together. If all the monomers, polymers, sugars, salts, enzymes, and vitamins of an organism were mixed together, some reactions would occur at random but would not be orderly enough to be considered life. Instead, cells are filled with many compartments, the organelles, each surrounded by its own unique selectively permeable membrane. Because the protein channels and pumps are made under the guidance of the nucleus, the cell can control the numbers and types of channels and pumps it makes and inserts into each type of organelle membrane. Organelles thus have membranes with different permeabilities or pumping capacities, depending on the instructions generated by the nucleus. Figure 3, 9 numerous organelles produce small vesicles that move through the cell, either remaining discrete for long periods or fusing with other organelles or vesicles. In order to fuse, two membranes must be quite similar in composition. Here, a single membrane bilayer is shown as a double line, one orange and the other black. Membrane dissimilarity prevents two compartments from fusing. For example, the nucleus does not fuse with mitochondria or chloroplasts. When a vesicle fuses with the membrane at the cell surface, its contents are deposited outside the cell and the vesicle membrane becomes part of the cell surface. The vesicle membrane and the plasma membrane have been drawn with the same colors to indicate their similar composition. Figure 3, 10 An impermeable membrane allows nothing to pass through, whereas a freely permeable membrane stops nothing. A selectively permeable membrane allows certain materials to pass through more readily than others, with the result that chemical concentrations on the two sides differ. This membrane is impermeable to substance A but permeable to B. Table 3, 3 Active transport in roots of corn. A freely permeable membrane, which allows everything to pass through quickly, would be rather useless for a cell, as would an impermeable membrane, one that does not allow anything through at all. The last important property of membranes is that they are dynamic, constantly changing. Cell membranes are not simply established and left unchanged throughout the life of the cell. Instead, new components are constantly being inserted and old ones removed. If the function of the cell is always the same, the old and new components are similar, but if the cell must change its function, newly inserted components are different from the retracted ones. As the nature of the membrane changes, the nature of the cell changes also. Table 3, summary of transmembrane movement. Basic cell types. At the most basic level, both cells and organisms can be classified as either prokaryotic or eukaryotic. Prokaryotic cells are simpler than eukaryotic ones and are found only in domains bacteria and archaea. It is hypothesized that prokaryotic cells represent the most archaic lines of evolution and that eukaryotic cells evolved from them. Eukaryotic cells, found in plants, animals, fungi, and protists, are more complex than prokaryotic cells. The most striking difference, the one that gives them their name, is the presence of a true membrane-bounded nucleus in eukaryotic cells. In addition, eukaryotic cells have many organelles that allow them to be more diverse and complex, both morphologically and physiologically. Plant cells. Although various parts of a plant, roots, wood, bark, leaves, and flower parts, appear to be quite diverse, virtually all of their cells have all of the following organelles. Exceptions are rare. As each type of cell develops, certain organelles may become modified and more or less abundant, but usually none is lost completely. Protoplasm. All cells, either prokaryotic or eukaryotic, are made of a substance called protoplasm. This name was given early, when it was thought that protoplasm was a distinct substance such as water, oxygen, or iron, and that one of its properties was life itself. We now know that protoplasm is a mass of proteins, lipids, nucleic acids, and water within a cell. Except for the wall, everything in the cell is protoplasm, composed of the following organelles. Figure 3:11 Generalized plant cell. Plasma membrane. The plasma membrane is the membrane that completely covers the surface of the protoplasm. Because it is the outermost surface of the protoplast, it must be impermeable to harmful materials and permeable to beneficial ones, therefore, it is selectively permeable. Molecular pumps in the plasma membrane actively transport needed molecules inward and pump others outward for secretion. Because one side of the plasma membrane faces the external environment and the other side faces the cell, the two sides are quite different, especially in the types of protein they contain. We know very little about the proteins and lipids that make up the plasma membrane because the membrane does not separate cleanly from the protoplast. Almost all attempts to isolate the plasma membrane for study have experienced considerable contamination from other parts of the cell. Figure 3:12 A healthy, growing cell. Its protoplast is pressed firmly against the cell wall, and thus, its plasma membrane is not visible. These cells have been treated with salt to draw water out of the cell, causing the protoplast to shrink. As a result, the plasma membrane is visible. Nucleus. The nucleus serves as an archive, or permanent storage place, for the organism's genetic information. This text is just a brief introduction to plant biology. No one can imagine the number of pages required to store all of the information necessary for building and maintaining a single cell. However, all of that information must be stored in the DNA inside every nucleus, and the storage must be safe and permanent. Information is useless unless it, or copies of it, can be retrieved and used. The nucleus carries out information retrieval by making copies of specific parts of the DNA whenever that information is needed. The copy is not DNA, but a type of ribonucleic acid called messenger RNA. An exciting area of research involves understanding how the nucleus, in response to signals from the rest of the cell, searches its DNA for the needed information and then makes copies of it without accidentally copying other similar information. The nucleus of a eukaryotic cell is always surrounded by a nuclear envelope composed of an outer membrane and an inner membrane. The nuclear envelope separates nuclear material from the rest of the cell, and it contains numerous small holes, nuclear pores. Involved in the transport of material between the nucleus and the rest of the protoplasm. Nuclear pores have a complex structure and exert control over the movement of materials. If a nucleus is extracted from a cell and placed into water, it swells. This can happen only if the pores prevent material from oozing out as the nucleus absorbs water. Prokaryotes have no nuclear envelope. Instead, DNA is simply mixed with the rest of the cell contents. Within the nucleus is a substance called nucleoplasm. Like protoplasm, this name was given before the composition was understood. We now know that nucleoplasm is a complex association of DNA, enzymes and other factors necessary to maintain, repair, and read DNA, histone proteins that support and interact with DNA, several types of RNA, and water and numerous other substances that are necessary for nuclear metabolism. Nuclear DNA is always closely associated with histones, and this complex of the two is known as chromatin. As a cell ages or its metabolic activities change, so do the nucleus and nucleoplasm. In cells undergoing rapid cell division, the DNA, histones, and duplicating enzymes may dominate the nucleoplasm. However, in mature cells that are not dividing, messenger molecules and reading enzymes may be more abundant. Figure 3.13a preparation called a freeze fracture.